So today I'm going to be discussing Heligosmoides, polygyrus induces TLR4 on mirroring mucosal T cells that produce TGFβ after LPS stimulation. So what the paper is looking at, so we have the worms, H, polygyrus, and they infect the mice. And what ends up happening is the worms are able to survive because they're able to evade the immune response from the host. So this paper is looking at how that's done and then hopefully being able to understand that and then relate that to Crohn's disease or IBD, something like that. So we start off by the paper looking at, there's a lamina propria mononuclear cells, the LPMC. I'm going to just only say LPMC from now on. The paper is available in the link. So you have the LPMC, and we want to see what happens when you have the LPS, lipopolysaccharide, and we have it induce the LPMC without any prior, like the mouse is not infected in any way before. So the worms are not even present on the scene. So we put an LPS and we see, and this is going to be looking at figure 1, and this is going to be B, C, and D. And we're looking at B, C, and D, and we see that B is looking at TNF alpha, and these are all looking for TNF from the supernatant that was used. So they take out the cells, the LPMC cells from the mice that was unaffected, put it on a plate, then they look at the supernatant, and then they see, and they put an LPS if they want, and they look at the supernatant afterwards and see what was different in the supernatant. And one of the things they looked for was TNF alpha. They ran a TNF alpha ELISA. No results. Nothing was conclusive. Barely detectable was actually the result that they came up with for TNF alpha. So what that means was the LPMC does not respond to LPS by producing TNF alpha. We look at IL-12. IL-12, the exact same results. We're looking at BD. Nothing happens as well. Therefore, LPMC does not respond to LPS by increasing IL-12. IL-10, a little bit different. There's actually an increase. So with LPS response from the LPMC is an increase in IL-10. And what's also not shown here is TGF-beta. TGF-beta also, it says somewhere, yeah. They did not. Colonization with H. polygyrus did not enhance LPS-induced IL-10 production. Okay, yeah. So what that means is, so we're looking at the uninfected LPMC first, and it had IL-10 upregulation. It had TGF-beta upregulation. Good. Now we're going to look at when the worms are infected, if the worms infect it, how does the LPMC change? And there was no upregulation, like increased upregulation for IL-10, only TGF-beta. So we're looking at TGF-beta, and we're like, wow, this is what's up. We're checking this out right now. So now we know that the worms upregulate TGF-beta in the LPMC of the intestine. And that's pretty much figure 1 in a nutshell. And so the next figure, I was adding on to it a little bit in the previous figure 1, but the figure 2 is looking at when you have infected with worms, how does the LPMC respond? What's upregulated? And we found out that TGF-beta was upregulated. So we now know that something in the LPMC, lamina propria, which is like the region of the cells, and the mononuclear cells. And so we're looking at that, and we go, okay, so this paper was asking, what in the LPMC is increasing the amount of TGF-beta? And in the paper, it looks like they just guessed right correctly, but probably they didn't. And they were like, probably the T cells, which, I mean, makes a whole lot of sense. So what they do, they took the LPMC, and they had the control, and then they had, well, the first one right here in figure 2A, they had the control, and then they had the H. polygyrus. And the control was, and this is just the difference between the LPMC without infected with worms and with infected with worms. So it shows a response that when the mouse has the LPMC, which is infected with worms, there's going to be an increase in the amount of TGF-beta when it is stimulated by LPS. Crazy stuff. But we look at figure 2b, and this is where it gets interesting. This is when they start looking at it and they see what exactly is in the LPMC that's affecting it. And we see that they isolate out the T cells from the LPMC. So there's T cell enriched LPMC, and then there's LPMC with a T cell like absence. And we see those, and we're going to detect how much TGF-beta was produced when the control versus the worms in response to the LPMC with the modification of if there's T cells or not. And sure enough, when you have the T cell enriched, the same response still happens. But when you have with the T cell absence in the LPMC, there is really no response. The difference between the cells versus the cells with LPS, there is no difference. I mean, right here we have our positive controls: an alpha CD3 antibody for CD3 and CD28, and CD3 is pretty much on all T cells. So what this was saying was that if there was T cells around, then it would respond to it. Therefore, it would attack it, and then produce more TGF-beta. Pretty much that's what it's coming down to in here. So 2b is saying that, oh, the T cells that are present in the LPMC are causing upregulation of TGF-beta when exposed to the worms, which induce an LPS response. Okay, so we now know that, here's the deal, we now know that the LPMC T cells, which we now can shorten as LPT cells, they're ones that are pretty much causing the upregulation, or they're responsible for the upregulation in TGF-beta. So now this paper is like, we can do one better than that. I mean, that's just T cells. What's really causing this upregulation in TGF-beta? And they go and look at it. They do a Western blot, and they see it, we're checking it out. And they're like, okay, let's do a knockout. Let's do a regular wild type. And so they have the knockout and they have the wild type in 3A. It's a simple Western blot right there. We have the TLR4, which is what they looked at. You have the CD3 gamma, and then you also have HPRT. Sorry. HPRT is a housekeeping gene that's going to be present in all murine cells or mouse cells. So no surprise there that it all was expressed in the Western blot. Congratulations. CD3 gamma, yes, that makes sense. There should be T cells present. There's no reason why they shouldn't be. TLR4, different story. What's up with the TLR4? So they had the knockout mouse, which was a knockout for TLR4, so there should be no TLR4 mRNA present. And bingo, they did a good job knocking that out, because there is no TLR4 present with the knockout mouse. Whether the knockout mouse was infected with worms or not with worms, which NW is no worms and W is worms. That's what the paper simplifies it as. And so they have the wild type, which is the second part of the Western blot when it looks down to it. And the wild type is broken up into no worms, and this trial is done twice, I think just for confirmation. And what happens is, for both times, the no worms did not have an upregulation in TLR4 mRNA. But the worm-infected LPMC definitely did show an increase in the amount of TLR4 mRNA present. And that's when you see the two lines under the TLR4. I don't know if you can see that. Yeah, kind of like, you can't really see them that well, but they're there. And it's pretty obvious. So the paper looks at that and it goes, okay, TLR4 is a big deal. It's upregulated. Wow. TLR4 on the LPT cells that are part of the LPMC, that's affected by the H polygyrase, and that's just how the train of thought works. So now we're looking at 3, and 3 has so much more information that's thrown at us. I mean, there's B, C, D, E, but let's look at exactly what's happening in B, C, D, E. All right, so we're looking at B. We're looking at 3B right now. And it is it's a fax, and it's looking at CD3, it's co-staining CD3 and TLR4 MD2 on top of the cells. And this is what they look like. And here's a node NW, which is no worms. and then there's W with worms. And this one, when you're looking at it, shows that there's no, what happens was, when you have worms inside the LPT cells, there's an increase in the amount of TLR4 MD2 mRNA produced on the membranes of the T cells. So what this is saying is, because the internal control is no worms, so you're looking at it, what is the basal amount of stuff that's produced on the receptors, co-staining obviously for CD3 and TLR4, and then it looks at, with worms, is there any difference? And yes, there's a difference. There's an increase in the amount of TLR4 receptors on the membrane. And TLR4 receptors is redundant because TLR is toll-like receptor. Regardless of that, there's more of them on the membrane. Now we look at D, and D is even trickier. Let's see if we can show you D. D, same thing. You have CD25 and you have CD3. As I said earlier, CD3 is present on all T cells. Not a big surprise that it should be present on, not that they should be present on them, and CD25 is present on activated T cells as well. And sorry, they're not looking at CD3. They're looking at CD4. They're looking at T helper cells, and they're looking at CD25, which is on pretty much all T cells. So yeah, so they're looking at CD25 and CD4, and we have the wild type with worms. So we have the worm infested. And we have the worm infested with another staining technique. So when we're looking at this and we see the worms, there's a cluster of all these types of cells that are present with the CD4 and the CD25. And now what the paper was looking at was, let's look at and see which cells, is there a specific cell, if we co-stain the two stains for CD25 and CD4, if we stained on top of that, for all the cells that had TLR4 MD2 on the membrane, which cells would those be? And sure enough, 3D shows that the cells that have a high level of CD4 are going to have TLR4 on the membrane. Because you can see that this plot right here, the second one, there's a little bit less because there's less cells that have the TLR4 present on the membrane, but the ones that do have a higher level of CD4 than this amount. The lines inside of it are just markers to show you where stuff is, so you can have a proper bearing. So for the quadrants, and there's a quadrant number ratios at the outside of it as well. But it's really important to go and look at it and see that actually higher levels of CD4 result in more likeliness that there's going to be TLR4 present on the membrane. And the last step. So what this paper was saying was, okay, look at this. We know that CD4, so it's CD4 cells, T cells that really have a high level of CD4 will end up having TLR4 present on the membrane. So TLR4 is definitely a required receptor in order to upregulate the amount of TGF-beta. But this paper makes sure that it was TLR4, because just because it works for one receptor doesn't mean that there couldn't be more receptors that also it worked for. So you should probably be knocking out TLR4, looking at the response and noticing, okay, that's right. When we knock out TLR4, there is no upregulation of TGF-beta in the cells because you're hypothesizing that TLR4 is a required pathway in order to produce the TGF-beta. And what did I just explain? I explained all of figure 4 right there. Figure 4, they do the knockout of the TLR4 and they see how it's going to be affecting the cells when they're presented with LPS, and sure enough, there is no increase.